Your blood-making stem cells, known as hematopoietic stem cells, usually prefer a calm, steady pace. Think of them like skilled workers in a workshop. They operate best when they can take their time, carefully creating the immune cells your body needs. But when life throws challenges their way, such as an infection or an injury, they immediately step up production. The downside is that too much stress over the long term begins to wear them out, making them age faster and lose some of their ability to bounce back. Now imagine what happens when those same hardworking cells are sent into space. In low Earth orbit, nothing feels normal. Gravity is gone, radiation is stronger, and biology begins to behave in ways we don't fully understand. NASA's earlier studies, including the well-known twin study, already revealed that space can alter human biology in surprising ways, changing DNA, shortening or lengthening telomeres, and sparking inflammation. But until recently, nobody had tested how space might affect the very stem cells that produce our blood and immune systems. That gap inspired researchers at the University of California, San Diego's Sanford Stem Cell Institute to join forces with NASA and Space Tango. Together, they launched the Integrated Space Stem Cell Orbital Research Center, or ISSCOR, and sent stem cell experiments aboard several SpaceX resupply missions to the International Space Station. Their goal was simple but groundbreaking: to see how blood-forming stem cells hold up in space, not just during the mission, but before and after as well. The answers came quickly. After only about a month in orbit, the stem cells began showing clear signs of aging ahead of schedule. They became unusually active, burning through their energy stores instead of resting. This hyperactivity made them less capable of producing fresh, healthy blood cells. At the same time, internal problems piled up. DNA damage increased, telomeres began to fray, mitochondria signaled distress, and even normally silent genes turned on, throwing the cells off balance. In short, the environment of space seemed to push these cells to age much faster than they would on Earth. Radiation appeared to play a big role. During the space flights, NASA measured radiation doses similar to what you'd get from a medical scan, like a CT or X-ray. While those doses might sound low, the combination of cosmic rays, microgravity, and the general stress of space travel created a much more harmful effect at the cellular level. The result was a perfect storm for stem cell stress. Yet there's a silver lining. When the space-exposed cells were returned to Earth and placed in a healthy, supportive environment, some of their damage began to repair itself. It's as if the cells had been through a tough assignment, but, given time and care, were able to recover. That discovery is encouraging because it suggests that astronaut cells may not be doomed to permanent harm, and that with the right strategies, recovery is possible. For the scientists leading this work, the mission is just getting started. Protecting stem cells in space is not just about astronauts today. It's about preparing for the future, when humans will travel longer and farther, perhaps to Mars or beyond. Finding ways to shield cells from radiation, spotting early warning signals of stress, and even developing drugs or genetic tools could make the difference between a risky mission and a safe one. What makes this research so exciting is that it goes beyond science fiction. With every mission to the International Space Station, researchers get a clearer picture of how space changes us at the deepest level. By understanding how to slow or reverse these changes, we're not just preserving health in orbit, we're laying the groundwork for humanity's next big leap into the cosmos.